हेलो फ्रेंड्स, आज सेल द यूनिट ऑफ लाइफ का पार्ट 2 पढ़ेंगे। इसके बाद पार्ट 3 आएगा, फिर चैप्टर कंप्लीट हो जाएगा। सेल वॉल, a non-living rigid structure called the cell wall forms an outer covering for the plasma membrane of bacteria, some protists, fungi, algae, and plants. Algae have cell wall made of cellulose, glycans, mannans, and minerals like calcium carbonate. In other plants, cell wall consists of cellulose, hemicellulose, pectins, and proteins. Cell wall, primary cell wall, thin elastic, composed of cellulose, hemicellulose, and pectin, gradually diminishes as cell matures, capable of growth. Secondary wall S1, S2, S3, rigid, thick, composed of cellulose, hemicellulose, pectin, absent in meristem cells. Inner side of primary wall toward cell membrane. Tertiary wall present only in tracheids of gymnosperm composed of hemicellulose and xylem. Mid-lamella, common layer between two cells. Mid-lamella is consists of calcium and magnesium pectates. Plant cement. Amount of calcium is more. Cellulose is a main constituent of cell wall, but addition to cellulose, hemicellulose, cutin, pectin, lignin, suberin are also present in cell wall. Cell wall worked as frame or protective layer of cell, exoskeleton of cell. Cellulose microfibrils and microfibrils are arranged in layer to form a skeleton of cell wall. In between these layers, other substances like pectin, hemicellulose may be present. These form matrix of cell wall. Primary wall, secondary wall, layer 1, layer 2, layer 3, cytoplasm. Cell wall. Cell wall materials, hemicellulose, pectin, lignin are synthesized in plants, Golgi bodies, or dicytosomes. Formation of cellulose occurs by cell membrane. Plasmodesmata. Name proposed by Straberger 1901. These are cytoplasmic connections between two adjacent plant cells. Plasmodesmata are characteristic of multicellular plants. Endoplasmic reticulum tubules, desmotubules help to maintain continuity of cytoplasm. The cell wall and mid-lamella may be transversed by plasmodesmata which connect the cytoplasm of neighboring cells. Functions of cell wall. Cell wall gives shape to the cell. It protects the cell from mechanical damage and infection. It also helps in cell-to-cell interaction. It provides barrier to undesirable macromolecules. Term cytoplasm. Cytoplasm was given by Straberger for the part of cell present between the nucleus and cell membrane. Cytoplasm can be divided into two parts, cytosol and trophoplasm. Cytosol, liquid matrix of cytoplasm except organelles. Trophoplasm, part of cytoplasm containing organelles and non-living inclusions due to plasm. Cell organelles. Metabolically active and living structure of cytoplasm are called organelles. Endomembrane system. Which each of the membranous organelles is distinct in terms of its structure and function. Many of these are considered together as an endomembrane system because their functions are coordinated. The endomembrane system include endoplasmic reticulum, Golgi complex, lysosome, and vacuole. Since the function of the mitochondria, chloroplast, and peroxisomes are not coordinated with the above components, these are not considered as part of the endomembrane system. Endoplasmic reticulum. Electron microscope studied of eukaryotic cells reveal the presence of a network of reticulum of tiny tubular structure scattered in the cytoplasm that is called the endoplasmic reticulum. Components of endoplasmic reticulum. Cisternae. These are long, flattened, and unbranched units arranged in stacks. Vesicles. These are oval membrane-bound structures. Tubules. These are irregular, often branched tubes bounded by membrane. Tubules may be free or associated with cisternae. Structure of endoplasmic reticulum is like the Golgi body, but in endoplasmic reticulum, cisternae, vesicles, and tubules are isolated in cytoplasm, and these do not form complex. Golgi body is localized cell organelles, while endoplasmic reticulum is widespread in cytoplasm. Endoplasmic reticulum is often termed as system of membranes. Endoplasmic reticulum divides the intracellular space into two distinct compartments. For example, luminal inside endoplasmic reticulum and extraluminal cytoplasm compartments. Rough endoplasmic reticulum, granulars, smooth endoplasmic reticulum, agranulars. Rough endoplasmic reticulum, 80 as ribosomes binds by their larger subunit with the help of two glycoproteins, riboflavin first and second, on the surface of rough endoplasmic reticulum, mainly composed of cisternae. Abundantly occurs in cells which are actively engaged in protein synthesis and secretion. For example, liver, pancreas, goblet cells. A smooth endoplasmic reticulum, ribosome and riboflavins absent, mainly composed of tubules. Abundantly occurs in cell concerned with glycogen and lipid metabolism. In animal cell, lipid like steroidal hormone and synthesized in smooth endoplasmic reticulum. For example, adipose tissue, interstitial cell, muscles, glycogen storing liver cells, and adrenal cortex. Nucleus. Nuclear pore, rough endoplasmic reticulum, ribosome, smooth endoplasmic reticulum. Modification of endoplasmic reticulum. Number first, sarcoplasmic reticulum. This smooth endoplasmic reticulum occurs in skeletal and cardiac muscles. Endoplasmic reticulum stores calcium and energy-rich compound required for muscles contraction. Microsomes. These are pieces of endoplasmic reticulum with associated ribosomal particles. These can be obtained by fragmentation and high-speed centrifugation of cells. They do not exist as such in the living cell. Scientists used microsome for in vitro protein synthesis study. Functions of endoplasmic reticulum. Number first, mechanical support. Microfilaments, microtubules, and endoplasmic reticulum forms endoskeleton of cell. Number second, intracellular exchange. Endoplasmic reticulum forms intracellular conducting system. Transport of materials in cytoplasm from one place to another may occur through the endoplasmic reticulum. At some places, endoplasmic reticulum is also connected to plasma membrane, so endoplasmic reticulum can secrete the materials outside the cell. Rough endoplasmic reticulum provides site for the protein synthesis because rough endoplasmic reticulum has ribosome on its surface. Lipid synthesis. Lipid, cholesterol, and phospholipids synthesized by the agranular portion of endoplasmic reticulum, a smooth endoplasmic reticulum. Endoplasmic reticulum also helps in the synthesis of lipoprotein and glycogen. Cellular metabolism. The membranes of the reticulum provides an increased surface for metabolic activities within the cytoplasm. Formation of nuclear membrane. Fragmented vesicles of disintegrated nuclear membrane and endoplasmic reticulum elements arranged around the chromosome to form a new nuclear membrane during cell division. Formation of lysosomes, Golgi bodies, and some microbodies. Detoxification. A smooth endoplasmic reticulum concerned with detoxification of drugs, pollutants, and steroids. Cytochrome P450 in endoplasmic reticulum act as enzyme which function in detoxification of drugs and other toxins. Endoplasmic reticulum provides the precursor of secretory materials to Golgi body. Golgi complex. Camellia Golgi 1898 first observed densely stained reticular structure near the nucleus. These were later named Golgi bodies after him. Golgi body, also named as lipochondria, rich in lipids. Dicytosome plants Golgi body. The cytoplasm surrounding Golgi body have fewer or no other organs. It is called Golgi ground substance or zone of exclusion. Structure. Golgi complex is made up of three parts. First, cisternae. These are flat disc sac-like structure. Many cisternae are arranged in a stack parallel to each other. Diameter 0.5 micrometer to 1.0 micrometer. Dense opaque material inside cisternae is called nodes. Varied number of cisternae are present in Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus. Convex surface of cisternae which is towards the nucleus is called cis phase of forming phase. Concave surface of cisternae which is towards the membrane is called trans phase or maturing phase. The cis and trans phase of the organelles are entirely different but interconnected. Number 2. Tubules. These are branched and irregular tube-like structure associated with cisternae. Vesicles. Transition vesicles and mature vesicles. Golgi apparatus. Cisternae. Vesicles fusing with cis phase. Tubules. Function of Golgi complex. Cell secretion. Chief function of Golgi bodies is secretion, export of macromolecules. Secretion involves three steps. Number first, Golgi body receives the materials from endoplasmic reticulum through its cis phase. These materials are chemically modified by Golgi body. For example, glycosylation of or glycosidation of proteins and lipids take place in Golgi body and its yields glycoproteins and glycolipids. Number C. After chemical modification, materials are packed in vesicles. These vesicles are pitched off from transphase of Golgi body and discharged outside of the cell. All the macromolecules which are to be sent outside the cell move through the Golgi body. So Golgi body is termed as director of macromolecules traffic in cell or middleman of cells. The Golgi apparatus principally perform the function of packaging materials to be delivered either to the intracellular targets or secrete outside the cell. Materials to the package in the form of vesicles from the endoplasmic reticulum fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. This explains why the Golgi apparatus remain in close association with the endoplasmic reticulum. A number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its trans phase. Golgi apparatus is the important site of formation of glycoproteins and glycolipids. Formation of lysosome. It is collective function of Golgi body and endoplasmic reticulum. Synthesis of cell wall material. Polysaccharide synthesis. Cell plate formation. Phragmoplast during the formation of acrosome during spermiogenesis, formation of male gametes. Lysosome, these are membrane-bound vesicular structure formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzyme, hydrolase, lipase, protease, carbohydrate, optimally active at the acidic pH equal to 5. These enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acid. With the exception of mammalian RBC, they were reported in all animal cells. In plant cells, large central vacuole function as lysosome. So in higher plants, lysosome are less frequent, but number of lysosome is high in the fungi. Periplasmic space between cell wall and cell membrane in bacteria may play a similar role. Lysosomes are spherical bag-like structures which are covered by single unit membrane. They are large-sized in phagocytes WBC. Lysosomes are filled with 50 different types of digestive enzymes termed as acid hydrolase for digestion of all types of macromolecules. These acid hydrolase function in acidic medium pH is equal to 5. Membrane of lysosome has an active H plus pump mechanism which produces acidic pH in lumen of lysosome. Lysosomes are highly polymorphic cell organelle because lysosomes have different physiological state. Types of lysosome. Primary lysosome or storage granules. These lysosomes store enzyme acid hydrolase in the inactive form. These are newly formed lysosomes. Digestive vacuoles or heterophagosomes. These lysosomes formed by the fusion of primary lysosome and phagosomes. These are secondary lysosomes. Residual bodies. Lysosomes containing undigested material are called residual bodies. These may be eliminated by exocytosis. Autophagic lysosomes or autophagosomes. Lysosomes containing cell organelles to be digested are known as autophagosomes. Functions. Intracellular digestion. Number first, heterophagy. This is the digestion of foreign materials received in cell by phagocytosis and pinocytosis. Number two, autophagy. Digestion of old or dead organelles. Autophagy also takes place during starvation of cell. Extracellular digestion. Lysosome of osteoclast bone-eating cells dissolve unwanted part of bones. Extracellular digestion also occurs by fungal lysosomes. Cellular digestion, autolysis. Sometimes all lysosome of a cell brush to dissolve the cell completely, or lysosome called as suicidal bag of cell. Old cells are removed, but autolysis unwanted organs of embryo are destroyed by autolysis. Cathepsin of lysosome digests the tail to tadpole of frog during metamorphosis. The vacuoles is the membrane-bound space found in the cytoplasm. It contains water, sap, excretory product, and other materials not useful for the cell. The vacuole is bound by a single membrane called tonoplast. In plant cells, the vacuolation occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitates the transport of a number of ions and other materials against concentration gradient into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for excretion and osmoregulation. In many cells, as in protists, food vacuole are formed by engulfing the food particles. Mitochondria. Number of mitochondria depends upon physiological activity of cell. One in Microasterias chlorella fusca alga. All the mitochondria present in a cell are collectively called chondriome. Usually, plant cells have fewer mitochondria as compared to animal cells. In higher animals, maximum mitochondria are found in flat muscles of birds. Mitochondria are differ in size and shape can make its shape sausage or cylindrical. Diameter 0.2 to 1.0 micrometer, average 0.5 micrometer, length 1.0 to 4.1 micrometer. Mitochondria is also named as powerhouse of cell or ATP mill in cell, cell within cell, mostly busy and active organelle in cell, semi-autonomous cell organelle, endosymbionts of cell. Structure mitochondria, unless specifically stained, are not easily visible under the microscope. Mitochondria are stained by genus green bee. Mitochondria is covered by double unit membrane. The outer membrane is smooth and inner unfolds into several cisternae. Christie. Outer membrane has more phospholipids, phosphatidyl choline, and cholesterol are compared to inner membrane. Phospholipid in inner membrane is mainly diphosphatidylglycerol, and inner membrane have more protein. The outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments. For example, the outer compartment and the inner compartment. The inner compartment filled with the dense homogeneous substance is called the matrix. The outer membrane form the continuous limiting boundary of the organelle. The two membranes have their own specific enzyme associated with the mitochondrial function. Both the membranes are separated by the space called perimitochondrial intermembrane space. Inner membrane is folded into a number of finger-like cristae. In metabolically active mitochondria, number of cristae is higher. Many electron carrier cytochromes are arranged in a definite sequence in inner membrane of mitochondria, which form electron transport system. Inner membrane is studded with pin head particles called oxysomes or elementary particles or F2 F1 F0 F1 particles or ATP synthesis. These particles first described by Ernst Moran. Head of oxygens or F1 is concerned with oxidative phosphorylation, formation of ATP by energy of oxidation. Mitochondrial matrix have enzyme for Krebs cycle aerobic respiration. Besides the enzyme matrix have a complete protein synthesis apparatus, ribosome 70S, DNA, few RNAs and enzymes. So mitochondrial cause as semi-autonomous cell organelles. Single, double strand and circular neck DNA present in mitochondrial matrix. Mitochondrial DNA is 1% of total DNA in a cell. It is rich in GC content. Mitochondrial DNA can code the synthesis of some types of proteins. Rest of the protein and enzyme of mitochondrial are synthesized under the control of nuclear gene. Enzyme for replication and transcription of DNA like DNA polymerase and RNA polymerase are found in mitochondrial matrix. Function of mitochondria. Mitochondria are site of aerobic respiration and ATP production. Outer membrane, inner membrane, matrix, cristae, intermembrane space. F1 particles, matrix, inner chamber, circular mitochondria, inner mitochondrial membrane, cristae, outer mitochondrial membrane, ribosome 70S. Biogenesis of mitochondria. New mitochondria arise from division of pre-existing mitochondria. Mitochondria divide by binary fission. Endosymbiotic origin from prokaryotic cells. Number first, type of DNA. DNA sequence double stranded circular GC rich. Number second, type of ribosome 70S. Number third, divided by binary fission. Plastids. Plastids are found in all plant cells and in euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting specific colors to the plants. Based on the presence or absence and type of pigments, plastids can be classified into chloroplast, chromoplast, and leucoplast. Types of plastids. Number first, chromoplast. In chromoplast, fat-soluble carotenoid pigment like carotene, xanthophyll, and others are present. This gives yellow, orange, or red color to the part of the plant. Chlorophylls either absent or occur in very less amount. Chromoplasts occur mainly in pericarp and petals. Red color of tomatoes is due to the red pigment lycopene of chromoplast. Chromoplasts occur in petals, but color in petals is mainly due to water-soluble pigment which are found in cell shape, for example, anthocyanin. Number 2, chloroplasts. The chloroplasts contain chlorophyll and carotenoid pigments which are responsible for trapping light energy essential for photosynthesis. Number 3, leucoplasts. The leucoplasts are the colorless plastid of varied shape and size with stored nutrients. Amyloplasts store carbohydrate, starch, for example, potato. Aleuroplasts store oil and fats, whereas the aleuroplasts store proteins. Pigment and lamellar structure absence in leucoplasts generally occurs in non-green and underground plant cells. Different type of plastids may transform from one form to other because genetic material is similar. Number, shape, and size of chloroplast. Majority of the chloroplast of the green plant are found in the mesophyll cells of the leaves. Number varies from 1% of the Chlamydomonas, a green alga, to 20 to 40% in the mesophyll. These are lens-shaped, oval, spherical, discoid, or even ribbon-shaped. Length and width are also variable. Length 5 to 10 micrometer, width 2 to 4 micrometer. Structure of chloroplast. Membrane like mitochondria, the chloroplasts are also double membrane bond. Out of the two, the inner membrane is relatively less permeable. Outer membrane contains porins. The space limited by the inner membrane is called the stroma. Structure of chloroplast. Lipid droplets. Outer membrane, inner membrane, stromal lamella, granum, stroma, ribosomes, starch granules. Section view of chloroplast. Components of stroma. Number A, thylakoids. In the stroma, a number of organized flattened membranous sacs are present called thylakoids. Thylakoids are arranged in stacks like the peels or coins called grana, singular granum, or the integral thylakoids. Each chloroplast contains about 40 to 60 granum. Stroma lamella are Frett's channel or stroma thylakoids are flat membranous tubules connecting the thylakoids of the different granum. The membrane of the thylakoid encloses a space called lumen. Chlorophyll, photosynthetic pigments are present in the thylakoid's membrane. A photosynthetic functional unit located in the liquid membrane contains of about 250 to 400 molecules of various pigments, chlorophyll A, chlorophyll B, carotenes, xanthophyll, etc. is called as contagium. Enzyme. The stroma of the chloroplast contains enzymes required for the synthesis of carbohydrate. For example, enzymes of Calvin-Cycle or Dark's reaction and protein synthesis. DNA. Stroma contain small double-stranded circular DNA molecules. Ribosome. The ribosome of the chloroplast are smaller 70 as than the cytoplasmic ribosome 80s. Chloroplasts have their own genetic system and complete protein synthesis machinery, double-stranded DNA, RNA, ribosome, enzyme, amino acids, but enzyme for photosynthesis are synthesized by both genes of nucleus and chloroplast. Thus, chloroplasts are also called as semi-autonomous organelles both of the cell. Function. Photosynthesis. The chloroplast trap the light energy of sun and transform it into the chemical energy in the form glucose. Biogenesis. Number first, from proplastid. Number two, from binary fission of pre-existing plastid. Origin, symbiotic origin by the cyanobacterium.